We're about ready to begin. Once again, good evening or afternoon to everyone who's joining us today for Dr. Doris Hendig's webinar on the role that modifier genes and biomarkers play in PXE. I'm Christine Vokey from PXE International, Director of Education and Support. I want to let you know a little bit of housekeeping details. If you have questions today on anything that Dr. Doris Hendig is telling us, you need to type your questions into the question pane that you will see on the right-hand side of your screen. You are all muted, so you cannot speak your questions. And if you have any technical problems, you can also type a question into that question pane. I will see all those questions and try to help you with whatever issue you're having. Our best suggestion is often to just try logging out and logging back in if you're having some problems, especially sound problems. So I hope everyone can hear and isn't having any problems. All right, with that, I'm going to introduce Dr. Hendig. Again, I'm Christine Vokey, Director of Education and Support at PXE International, and I'm very excited today to have another great contributor to PXE research with us, Dr. Doris Hendig. She is a research fellow at the Institute of Laboratory and Transfusion Medicine of the Heart and Diabetes Center in North Rhine-Westphalia in Bad Oeynhausen, Germany. She received her PhD in biochemistry in 2008 from the Bielefeld University in Germany. One of her major research interests is in clarifying the pathology of extracellular matrix changes that cause disease in humans, and one of those diseases of interest is PXE. The PXE research group in Bad Oeynhausen is analyzing PXE causing sequence variations in the gene. They're looking for biomarkers for PXE that can be used to monitor the progression of PXE, and they're interested in identifying secondary genetic risk factors that may contribute to the severity of PXE. Dr. Hendig's work on PXE was recognized by the American Association of Clinical Chemistry and by the Southwest German Association of Dermatologists in 2005, and her research on PXE has been funded by grants from German research organizations. Her webinar today is titled, What Role Do Modifier Genes and Biomarkers Play in PXE? And with that, Dr. Hendig, I welcome you to PXE International's webinar, and thank you for being here.
Yeah, thank you very much, Christine, for this nice introduction and the great opportunity to present a webinar. And I also like to welcome all the audience, my dear ladies and gentlemen, and dear colleagues. In the next 30 minutes, I would like to present some aspects of our work. And as Christine already said, the title of my webinar is What Role Do Modifier Genes and Biomarkers Play in PXE? First of all, I would like to give you a short introduction about the causes of PXE, why people or patients suffer from PXE. What we all know that PXE is a genetic disease. So Terry, I would like to switch to the next slide, please. Okay. What I think most of you know is that PXE is caused by mutations in ABCC6. ABCC6 is a member of the great ABC transporter superfamily. ABC stands for ATP ATP binding cassette, which means that these proteins need ATP. ATP is an energy source for transporting a variety of metabolites across barriers, for instance, across the cell membrane. ABCC6 has also, or is hypothesized or postulated to have a transport function to serve as an efflux pump, but we researchers do not know what this protein is currently transporting. To understand why we investigate modifier genes, we must first have a look at the reason for PXE manifestation. On this slide you see a scheme of the protein, which is located in the membrane of, for instance, hepatocytes. Hepatocytes are cells from the liver. And what you can also see on this slide is that over the proteins there are red dots, and these red dots are changes in amino acids, also changes in substances of this protein which are mutated, and as you already or moreover you can see that these changes are across all regions of the protein. Next slide, please. What function is proposed for ABCC6? I tried from my experience with other patient organizations. That's quite difficult to describe what is the function of this protein. I just made a picture in this case where I want to show that these questionnaires should be this unknown factor, what we think is transported by ABCC6. We currently do not know what kind of metabolite, what kind of molecule this unknown factor presents. I've made ABCC6 as a crane or as a pump, which is located on or between a barrier, and we think that the ABCC6 is transporting these question marks across this barrier to the outside, for instance, of a cell. Can you switch to the next slide, please? Next slide, please. Yes, thank you. When ABCC6 is damaged through mutations, or when ABCC6 function is lost, we proposed that this unknown factor is accumulating in the inside inside of the cell, and that on the outside there's a lack of this unknown factor. And in consequence, elastic fibers, which are normally nearly located to barriers where we think ABCC6 is located, that there this mineral crystals deposit in or besides these elastic fibers, and consequently they damage them, so they get broken, and then there is damage to the tissue structures. Next slide, please. A short summary of the genetics of PXE. PXE is an autosomal recessive disorder. What we know is that more than 390 different ABCC6 mutations cause PXE and have been identified so far. All of them will lead to a loss of ABCC protein or its function. And what's quite interesting is that that the mutation detection rate is about 90%, but that means also that about 5 to 15% have only one or no ABCC6 mutations. So when we summarize here and we say, when we already know what's the cause for PXE is, and what's then the reason for studying modifier genes or looking for biomarkers. Next slide, please. When you look at PXE patients, you will see that there's a high phenotypic variability. I'm not sure if any one of you knows the term phenotype. Phenotype means the expression of the disease. If or how severe are the eyes affected, how severe are the skin lesions, is there anything or is there trouble with the blood vessels? This is called a phenotype or expression of disease. It's quite interesting that PXE, it's a monogenic genetic disease. That means it's caused by one gene defect. But when you look at patients who carry the same mutations, you see that there is a high inter-individual variability, even in families with more cases. We have, for instance, such a family in Germany. There are 3 brothers affected, but they are all present with a quite different expression of the disease. The one has a more severe eye involvement, and on the in the other boy, for instance, you see that there's typical or expression of the skin lesions. So this raises the question if there's an involvement of other environmental factors, for instance. That means nutrition can play a role, hormones, lifestyle, but also modifier genes or genetic interactions. And that was the reason to look at so-called modifier genes, which might affect the expression of PXE, for instance. Next slide, please. So let me first define you what we mean when we talk about modifier genes and later biomarkers. A genetic modifier is when expressed able to alter the expression of another gene, for instance. Modifier genes can affect transcription, alter the immediate gene transcription expression, or they can affect phenotypes at other levels of organization by altering phenotypes at the cellular or organismal level. This is a definition from Nadeau in 2001 in a review in Nature Genetics. And I would also like to give you an example from another disease, thalassemia, where beta-globin synthesis is defective. Here also patients have diverse clinical characteristics and a variable expression of the disease. These patients present with a profound anemia and the requirement of regular blood transfusions. And when you here look at patient scores, you see that there are people who carry the same mutations but who have mild or undetectable symptoms. Recent studies or several studies with patients in this with beta thalassemia, for instance, show that there are numerous modified genes, that other genetic loci which were identified to influence or to affect the production of globins, the protein which is defect in this disorder. Next slide, please. So from the upcoming knowledge in research, it's becoming more difficult to have a strict decision between monogenic disorders and multifactorial or complex disorders, and this decision or this strict decision becomes more and more blurred. That means that the monogene is not only responsible for disease manifestation and the phenotype of the disease. Moreover, the genetic background contributes to mild or severe disease progression. So we asked the question if manifestation, severity, and progression of PXE is also influenced by modified genes, and we have done a lot of work on that in the last 5 years. Now I introduced you in modifier genes. Just let me introduce you in what we think or what we mean when we talk about biomarkers. I just copied the definition from Wikipedia, as I think it is quite easy to understand. A biomarker generally refers to a measured characteristic which may be used as an indicator of some biological state or condition, or here a disease. The term occasionally refers to substance whose presence indicates an existing living organism, and biomarkers are often measured and evaluated to examine normal biological processes, pathogenic processes, pharmacological responses to therapeutic intervention. And all of you mainly, I think, know commonly used blood test in medicine. So cholesterol is a biomarker in metabolic disease. The prostata-specific antigen is a biomarker in cancer, and C-reactive protein, for instance, is a very important biomarker in inflammation. So biomarkers are used in many scientific fields for better understanding of the biochemistry of a disease. So we are interested to uncover a biomarker for PXE, for instance, to have a better and earlier diagnosis, for instance, or biomarkers, moreover, could serve as markers of disease and for evaluating harder to reach tissues and organs, as biomarkers are normally more readily accessible, unlike non-invasive techniques. Next slide, please. So in this slide, I try to visualize where we could find such modifier genes, and when we detect modifier genes, we know that, or we hope that these genes could serve as, or the products of these genes could or might serve as biomarkers for PXE diagnosis or for disease monitoring. Unfortunately, we cannot measure ABCC6 because this is a protein which is located in cell membranes, and we don't have access to this, I think, source. So what could serve as biomarkers? It would be very nice if we could measure the unknown factor, but as we don't know what this factor is, we cannot try, or we are not able to measure it to have a look, or to say that this disease is, for instance, PXE. What biomarkers can be then are factors that are influenced by the loss of ABCC6 function, that may be factors that accumulate inside of the cell, that are synthesized, or which synthesis is down or upregulated by accumulation of the unknown factor. That could also be on the outside of the cell, where where factors could be upregulated by the lack of these unknown factors, for instance, or downregulated. Factors can be proteins. Factors can be, for instance, answers of the cell, or activated or inhibited pathways. And when we come back to the modifier genes, we could also say what can be influenced by ABCC6 loss. That are factors, for instance, which serve to bind mineral crystals. This is a very characteristic hallmark of PXE. Modifier genes could also be factors that serve to synthesize ABCC6, that produces these unknown factors. ABCC6 just transport it. Can be factors that deliver energy, which is needed, for instance, when you think that one patient has only one ABCC6 mutation, what happens when there's not enough energy supplied in the cells. And these all together is quite complex, but may all affect the disease outcome, for instance, in PXE. Next slide, please. So the purpose of our study was to investigate factors that led to phenotypical variability in PXE. This phenotypic variability could be influenced by environmental factors, as I already pointed out on another slide or some time ago. Could be also caused by other genes. This is something I don't want to go in detail today, and we concentrated on the role of modifier genes. By searching for modifier genes for PXE, we investigated the frequency of sequence variants which are known to have a functional relevance, and which are known to modify the pathobiochemical processes seen in PXE. For instance, calcification, cellular stress, or angiogenesis. Next slide, please. Next slide, please. Okay, thank you. What are characteristic hallmarks of PXE? When you look at a biopsy from skin lesions, you see that there's a severe remodeling of the extracellular matrix structures, broken and damaged elastic fibers and collagens, for instance. Calcification could be detected, deposition of mineral crystals. When you have a look at the involvement of the eyes. There's a severe angiogenesis in mostly, I think, 90% of the patients, which led to a loss of vision if it's if disease is very or there's a high, severe progression, I think. And we know from the animal model and from cell culture studies and all over, moreover from measuring some factors in the blood of PXE patients, that there is an extreme or a severe oxidative stress. I just pointed out on the right side some factors which are known to lead to pathological calcification. I've summed them up. It's about, I find, you will find more than 50 different reasons or contributors which modulate or inhibit or lead to pathological calcification. There are inherited factors on the upper side of this figure on the right side. Mean there are several gene products which play a very important role in this context of arterial calcification, for instance. You have physiological arterial calcification inhibitors, where ABCC6 is now also listed in this kind of important proteins in this pathways. You have modulators of arterial calcification. This might be proteins which affect the structure of matrix structures, for instance, that might or might be prone to bind more crystals. And you have environmental and host factors, which are also very important, like is there a predisposition to diabetes, to chronic kidney disease, what is about aging, what is about hypertension, environmental factors like nutrition play a role, smoking plays a role, and also the taking drugs, for instance. These are all factors which contribute to arterial calcification. And we picked out some of them and looked, okay, what's happening to them in PXE. Next slide, please.
Doris?
Yes?
I have a question from a participant. If you could briefly review where the ABCC6 gene is most active, which cells is it most active in?
From the research we know in the hepatocytes, in the liver. No problem.
Thank you.
Okay, come back. Fetuin-A is the most important systemic calcification inhibitor, what we know to date. This protein has 3 different domains, as you see on the slide on the left side, which all can bind mineral crystals. And several Fetuin-A molecules together can bind these mineral crystals and build so-called calciprotein particles, which is shown on the right side of the slide in figure which is pointed by E, for instance. These calciprotein particles are normally cleaned out of the circulation so that they could not be binded by tissue structures, for instance, so that there is no crystal growth and the tissue is not got damaged. If Fetuin-A is missing, this can lead to a formation of crystals, as here seen on the on the downside of this slide. If you have a small crystal and you don't have Fetuin-A, then this crystal grow and could damage these tissues. Next slide, please. We asked if Fetuin-A is a biomarker, or what we know from the literature is that Fetuin-A is a biomarker for pathological calcification. And we looked, okay, we know that PXE patients have deposits of mineral crystals in several tissues, in several organs. What is Fetuin-A? Could it serve as a biomarker also in PXE patients which present with this characteristic pathological calcification? And we could detect that PXE patients have reduced Fetuin-A serum concentrations. And when you compare them to relatives or, for instance, healthy blood donors, which here serve as our control. And a study from Junio Ito and Jiang and Lili from Philadelphia, they also looked what is the Fetuin-A serum concentrations in the ABCC6 knockout mouse, which serves as an animal model for PXE, and they could also see that that Fetuin-A serum concentrations are reduced. We think that the reduction comes therefrom that Fetuin-A is bound to mineral deposits in the tissues, and therefore we could propose Fetuin-A as a biomarker to detect pathological calcification and maybe to monitor disease progression, as we know that Fetuin-A concentration decrease during disease progression. One problem is that this is not special or not typical for PXE. It's typical for all conditions which present with pathological calcification. Next slide, please. When we now go back to modifier genes, I would also point out that Fetuin-A serum concentrations are determined by the genotype. There are 3 different genotypes found in human beings, and all these 3 different genotypes correlate with high as Fetuin-A genotype 1 and low Fetuin-A serum concentrations as called Fetuin-A genotype 2. We did not find any association between the single Fetuin-A sequence variants with manifestation or disease progression, but we see that patients who carry the Fetuin-A genotype 2 have the lowest Fetuin-A serum concentrations. And therefore we postulate that this might be a risk factor for severe calcification due to reduced calcification inhibition capacity. Next slide, please. The next gene we have to look, we looked on, or we suggested as a probable candidate gene for modifying PXE progression is Osteopontin. Osteopontin is a regulator of biomineralization, which acts mostly locally or in the cell or in the soft tissue, and we also know that the expression and activity of this protein is actively regulated, as shown, for instance, in this figure. Here you can see that many different factors contribute to regulate this process of biological calcification, and that many metabolites here in the slide, for instance pyrophosphates or for instance the expression of transcription factors like RUNX2, are actively involved in regulating expression and activity of osteopontin. So next slide please, Terry. Thank you. As osteopontin is predominantly a transcriptional regulated gene, and the promoter of osteopontin is highly conserved among the human and other animal beings. We looked at polymorphisms in this promoter region, which were known to influence the expression or to affect the expression of osteopontin. And we looked at several different sequence variants and genotype then in PXE patients and healthy control probants, which were age and sex matched. And what we found is that 3 of these variants were appear to be significantly more often or more common in PXE patients. And one of them is quite interesting. It's an insertion in the position -155 and 156, which creates a second binding site for a very important transcription factor 1x2. And we suggest that if one patient carries 2 of these binding sites, that he has a much higher osteopontin expression, and that might also contribute to much more severe expression of oral disease. Because we know that if a pathological calcification is induced, for instance, like from other factors, that osteopontin is highly upregulated, and if you are further predisposed to carry such a variant, then it might be that your expression of osteopontin in the context of pathological calcification it's much more pronounced. Another factor is, when you come to the next slide, please, is oxidative stress. I think you all will have heard about that in I think in film breakings, that oxidative stress can be caused by many different factors, as shown in the left figure. Might be sun, might be smoking, might be environmental toxins, for instance. Oxidative stress leads to aging of structures, as I have shown here in the middle, with a car which goes older and older and older, and sometimes it will not go or work anymore. And to prevent oxidative stress, there are antioxidants, and for instance, these are found, for instance, in fruits and vegetables, and prevent this oxidative stress. I think that is also known in the population. Many of the alterations we observed in PXE patients in the cell culture models or in the animal model could be explained by oxidative stress. And when we postulate the accumulation of a factor, for instance, inside of the cell, we could think that hard accumulation of factors might be waste and might lead to oxidative stress for these cells. And this further leads to the remodeling of extracellular matrix structures, for instance. Next slide, please. We also looked at antioxidative enzymes, and antioxidative enzymes prevent oxidative stress, and there are 3 genes which are very important in human beings, that are the genes catalase, glutathione peroxidase, and the superoxide dismutase. And what we found here is that there's a correlation between genotype and the age of disease onset, so that we can say that if a patient carries one of these variants, for instance, has a much earlier onset of PXE, because if he carries this variant, the antioxidative enzyme don't works so as it is working as in other province or other people, and so it could not prevent the oxidative stress, and that might lead to a very early induction of PXE. What was quite interesting is that there's a correlation between a higher count of risk alleles, also associated with an earlier disease onset, but which also makes sense if you think that, for instance, all 3 antioxidative enzymes do not work very well. Okay, next slide, please. There's also another candidate gene where we have a look on in the context of angiogenesis. This is a growth factor which is very important in the growth of blood vessels, the vascular endothelial growth factor, VEGF. This growth factor, VEGF, is involved or associated with neovascularization, for instance in age-related macular disease. And what we investigated, that is also associated with the retinopathy we see in PXE patients. And from the current knowledge, it may be concluded or you know that anti-VEGF therapy is beneficial for the treatment of this neovascularization and could help to prevent the loss of vision. And therefore it seems justified to assume that that polymorphisms leading to altered VEGF expression may modify the severity of PXE retinopathy. Next slide, please. Oh, sorry. Okay, here you can see the typical retinopathy which we find in PXE patients. This is the neovascularization. Next slide, please. We investigated several polymorphisms in the VEGF-A gene, which is the gene coding for VEGF, the protein which affects this angiogenesis, and we detected that 5 VEGF-A sequence variants show a significant association with severe retinopathy in PXE. There was almost one allele, the -460 T allele, which was associated with increased vascularization and is therefore proposed to lead to severe retinopathy. And we think that the genotyping of VEGF-A gene polymorphism might prove useful as prognostic marker for the development of PXE-associated retinopathy. And that might need to improved or monitored anti-VEGF treatment. And this is currently one study which we investigate with our cooperation partners at the University of Bonn. Okay, next slide, please. Okay, now I would like to summarize that we have looked on several genes which might affect PXE manifestation or disease severity. The gene products of them might serve as biomarkers as if they are suitable for measurement in blood, for instance for Fetuin-A, VEGFA, Osteopontin, and several other things. But we did not found at present or until now a factor which is very typical for PXE. You can measure alteration in several diseases, so a change in VEGF serum levels, or for instance a change in Fetuin-A serum levels, is not characteristic for PXE, but it's characteristic for diseases which come along with pathological calcification, with severe remodeling of the extracellular matrix structures, and so on. But I think they might be useful for monitoring disease progression or, for instance, for therapeutic interventions. Okay, next slide, please.
Doris, we have
yes?
another question. If you could go back to that slide.
Yeah,
are these modifier genes, are any of these definitively associated with the expression of ABCC6 yet, or related to PXE, or you're just still
yeah, they are, yeah, all of these are related to PXE. This is what we have identified so far and also already published. I just picked out some of them. I think.
Okay, and how many are there now that you have identified? How many modifier genes?
I think 13 about 13. I think the problem is that genetic background is so different between human beings that you have to study big cohorts to what I want to say. We have studied a big patient cohort, about I think between 100 and 200 PXE patients, and we all see sequence variants in these genes are most often or more often common in the patient's group than compared to a control group, and it's quite difficult. Some of them are quite good to correlate them to a phenotypic expression. For instance, VEGFA was a quite interesting study, because we divided the patients cohorts in patients who have a severe retinopathy and patients who do not have such a severe retinopathy, and we see that the patients with a severe retinopathy have this one variant more often, or are more often carriers of this one variant. So this was quite an interesting results. Where we have a little bit problem is that the phenotyping of PXE patients, or to the measurement of the expression of PXE in patients, is it's not so easy. It's very difficult. There are some studies which have how do you call them, set up diagnostic criteria, but it's quite difficult to use them, because the medical consulting is also not very objective, I think, and that might be a problem to have a good correlation to a detailed phenotype, for instance. Okay, let's come to the last slide. Okay. Here I just want to summarize that the search for modifier genes is difficult, but I think it's worth being performed in view of a better knowledge of biological pathways which might be affected by the disease PXE, for instance. Our identification of modifier genes often leads to studies where other groups looked on the gene products of these modifier genes, and we could see then in animal models or in the cell culture models that the modifier genes also really play a role in the context of PXE, that the expression of these genes is affected by the loss of ABCC6 function, but at present we don't know where are the connection between is, because we don't know this factor which is transported by ABCC6. But I think that the identification of modifier genes for PXE underscores the importance of the analysis of gene-gene environment interactions and for understanding the development of complex phenotypes. I think it's also PXE is a complex phenotype because it's affected by so many different factors. And as we know that genetic modifiers are known to alter course and expression of disease, their gene products really might be interesting targets for monitoring the disease progression, or as well as for therapeutic intervention. And so what is very important, what we need in future, is that consistent phenotype measures that cannot be overemphasized, that we need that. Okay, and then I would like to come to my last slide. How do we find further modifier genes or biomarkers? I think in the last years there was many new technologies coming, which we all know from the term omics. Genomics is that kind of tool what predisposes or what tells us what can happen in our patients, for instance, because genetic backgrounds affect many pathways, and Then there are the transcriptomics, which tell us what appears to happening, what is really happening in cells, in organs, in tissues. The transcripts lead to the gene products, which are, for instance, ABCC6 or Osteopontin, Fetuin-A at the protein level. They play a role in what really happens in the cell. And a new field what's coming up in the last years is the metabolomics, which can tell us something about the metabolic state of a cell, for instance, of a tissue, of an organ. And currently, or from recent study, we know that there are more than 1 million metabolites which you can detect in human beings. And I think it's going to be interesting to use these technologies in monogenetic diseases, for instance, like PXE. And now I finished my webinar, I think. Last slide. I would like to thank all the people who helped us with the studies that are, for instance, all our German PXE patients. There I would like to thank you, Peter Hof, who's the chairman of the patients organization in Germany, my cooperation partners in the University of Bonn, the ophthalmologist, and Professor Sliska, who's the dermatologist for all PXE patients in Germany. And all this work could not have been done by funding, and this is also very important, and I'm glad that we have this funding for our research. And I thank you, my listeners today, for your attention.
Thank you very much, Doris. We still have a few questions, if you don't mind.
Yeah, no problem. I try to answer them.
Okay, thank you. We have a question that's kind of a basic one about PXE. Is there a relationship between mutations in ABCC6 and PXE symptoms and signs?
No. I think several groups try to establish a phenotype-genotype correlation, but it's not I think, successful in the years. There's no correlation between a genotype of PXE and a special phenotype. What we know is that a patient carries 2 ABCC6 mutations, the same or 2 different, what we suggest compound heterozygity, then this will lead to a PXE, for instance.
Okay. And then related to the modifier genes, is it enough to show that they're associated or correlated with PXE signs, and are you able to go further and show that a modifier gene causes a specific or sign in PXE or symptom?
I think a good example might be VEGFA, because this was shown in other diseases, not yet in PXE. We tried that, but it doesn't work. I'm sorry. Not everyone has, or it's difficult to have a look on the eye in PXE. I think what we know from AMD, for instance, or from age-related macular degeneration, is that some of these or many of these patients have increased VEGFA expression, and this is also correlated with sequence variant, which we also investigate in PXE patients, and this sequence variant correlate with a higher VEGFA expression. And when you think further, or when you look in AMD, for instance, you see that a higher expression of VEGFA leads to a higher or to a much more increased angiogenesis. So there you have a correlation between a phenotype and a modifier gene. That if you have a higher VEGFA expression, you have a stronger angiogenesis. If you have a stronger or severe angiogenesis, you have a much earlier point to, for instance, to lose your vision, for instance. And what we are currently investigating is if patients, for instance, who carry these mutation, which is also associated with a higher VEGFA expression, if they need much more improved VEGF or anti-VEGF-A treatment if you have to shorten the time to give them an injection, for instance, compared to other patients. This might be interesting if you think at the pharmacogenetic level in this case, for instance. It doesn't, yes.